Science, skills, and safety. In this lesson, we're talking about the scientific method. So what is a scientific method? The scientific method is a process of figuring something out through experimentation. Within the scientific method, we're going to hypothesize, we're going to come up with procedure in order to test that hypothesis, we're going to collect data, and then we're going to come to a conclusion. There's 8 main steps to scientific method: questioning, controlling variables, making hypothesis, planning, performing, observing, analyzing, and then finally discussing. So we're going to go through each point individually. Number 1, introduction and question. Within the introduction, relevant observations, inferences, background knowledge is provided. It's similar to brainstorming. You're coming up with an idea and you're thinking about that particular idea. The question that you're going to be looking at should be semi-formal. It should interest you and it can be about anything. So for instance, are taller people better at basketball than shorter people? Do 10 novels weigh more than 5 textbooks? These are all things you could test using the scientific method. Controlling the variables. Variables are factors that affect the outcome of an investigation. There's the independent variable, which is the variable that's changed. The dependent variable, which is the variable that's affected by the independent variable. We say that it depends on the independent variable. There's our controlled/standardizing variables, which are variables that remain unchanged and do not affect the outcome. This could be like if you're doing a test in a room, your controlled standardizing variables would be the temperature of that room. You don't want it to get too hot or too cold, and you don't want that to change between the different experiments because that could affect the outcome. And then we have the control group, which is the group that's not changed. It's normal conditions, and it's used to compare results. In many drug trials, the control group is usually the placebo group, where people think they're getting the medication, but they're actually not getting anything. What I'd like you to do is pause the video and determine the independent and dependent variable for these 3 questions. Keeping in mind the independent variable is the variable that's changed, and the dependent variable is the variable that's affected by the independent variable. So pause the video and try for number 1, 2, and 3. So for number 1, an archer tests a bow by seeing how tightening the bow changes how far the arrow can be launched. So in this case, the independent variable is the tightness of the bow, and the dependent variable is the distance the arrow travels. Number 2, a student experiments to see if the length of a pendulum affects how fast it swings. A pendulum, if you're unaware, is like a ball on a string that's fixed to a point, and it's going to move from point A to point B, and then back and forth. So in this case, the independent variable is the length of the pendulum, and the dependent variable is the speed of the swing. The speed of the swing depends on the length of the pendulum. Number 3, a boy tries on different running shoes to see how much each one slides on ice. The independent variable is the different shoes. That's the thing being tested. And the dependent variable is the sliding ability. The sliding ability depends on the shoes that are being worn. So again, I want you to pause the video and practice using this example case study here. I want you to find the independent variable, the dependent variable, and the control group. So in this example, the new sauce is the independent variable. That's the thing being changed. The dependent variable is the gas production. The gas production depends on the sauce being used. And group B were the control group because they weren't giving the new sauce that they thought they were. Once you have your question and your variables figured out, we can come up with a hypothesis. A hypothesis is a tentative answer about the outcome of an experiment. It should be presented in the if-then statement. It should indicate a cause and effect relationship for your dependent and independent variables, and it can usually be derived directly from the original question. It's an educated guess. It should only be a single sentence, and it must be feasibly tested. Now, for example, if the independent variable changes in some way, then it will affect the dependent variable in some way. So we see we have our if and our then. So we're saying if the independent changes, then the dependent variable will be affected in some way. So for instance, if someone is taller, then they will be better at basketball. So I want you to pause the video and create a hypothesis for each of the following 4 examples. Now, this isn't the only way they can be written, but your answer should follow a similar pattern to these examples here. Human age and the chance of getting heart attack. The hypothesis would be, if someone is older, then there's a higher chance of them having heart attack. Now, you could also say, if someone is older, then there's a lower chance of them getting heart attack. There's no definitive answer here. There's no right answer. This is what you are testing. So in this test, we would look at older people, and we would see if they have a higher chance of getting heart attack. If this was our hypothesis, we'd be proven right if older people got more heart attacks, and we'd be proven wrong if older people did not get more heart attacks. Skin cancer and the amount of UV light. If you are exposed to more UV light, then you'll have a higher chance of getting skin cancer. Again, a correct answer would also be, if you are exposed to more UV light, then you'll have a lower chance of getting skin cancer. We're not talking about the science behind it. We're talking about an example hypothesis for this particular question. You could also say it as, if you are exposed to less UV light, then you'll have a lower chance of getting skin cancer. That's also correct. Leaf color change and temperature. If the temperature decreases enough, then the leaves will start to change color. Amount of sleep per night and student test scores. If you get more sleep, then your test scores will improve. You could think of it the other way. Maybe you think that if you get less sleep, your test scores will go up because you'll have more time to study. In that case, you'd say, if you get less sleep, then your test scores will improve. Or if you get more sleep, then your test scores will decrease. So you can see we're taking our guess and we're putting it in the if-then statement. Our guess could be anything depending on what we think is going to be the outcome of the experiment. So we've come up with our question, we've identified our variables, we've written our hypothesis, now we need to plan our experiment. First thing we're going to do is make a materials list using bullet points, and then we're going to write a clear procedure. The materials list is pretty basic. You're going to use bullet points to list all the things involved. The procedure, however, is a little more complicated. A procedure is a sequential list of steps that are to be followed exactly during an experiment. Steps should be in order and numbered. You need to be specific and exact so that someone doing the experiment again later on could follow your exact steps and do the exact experiment. So what I'd like you to do is I want you to pause the video and write a procedure for washing your hands, starting from dry hands and ending with dry hands. Be as specific and exact as you can. Pretend the person you're speaking to has no knowledge of how to wash their hands. Once we have our procedure, we're able to start our experiment. When performing the experiment, we need to follow the procedure exactly. If the procedure is unclear, you need to ask. You need to be safe. Record which variable is being changed and how. Repeat the experiment a minimum of 3 times. This is to make sure there's no errors. So if you do something slightly different one way, maybe that'll impact your overall results. If you do it 3 times, you can get a much better idea of what your results should be. You should make careful notes of everything that occurs during your experiment, in addition to filling in charts and tables, and note possible sources of error. And this leads right into observing and recording data. This will be the data that we observe and record during the procedure. So we're going to record all data/observations in an appropriate form. This is generally in the form of charts and tables, because they're the easiest to read and fill in as you go. Record all data as clearly and accurately as possible, and make careful notes of everything that occurs during your experiment. Note possible sources of error in the experiment. 7. Analyzing results. When possible, create graphs to represent numerical data as it's easier to read and understand for others. Each table and graph contains a caption. So for instance, table 1 would be called table 1. Graph 1 would be called figure 1. The second graph would be called figure 2, etc. Identify patterns and trends in results and state what occurred / was found by referring to the tables and graphs. Do not discuss the results, just state them. According to figure 1, this happened as time went on. According to chart 2, this happened as we progressed through time, etc. So how do we graph? Rules for good graphing. Number 1, always give your graph a title in the following form. The dependence of your dependent variable on your independent variable. How height impacts basketball ability. That could be our title. Let's say you're doing a graph where you're studying the effect of temperature on the speed of a reaction. In this reaction, the speed will depend on the temperature. Therefore, speed is the dependent variable and temperature is the independent variable. As a result, the title of your graph could be the dependence of reaction rate on temperature. The x-axis of your graph is always your independent variable, and the y-axis is your dependent variable. For the graph described above, temperature would be on the x-axis, the one on the bottom of the graph, and reaction rate would be on the y-axis, the one on the side of the graph. So this would be reaction rate here, and on the x-axis we'd have temperature down here, and we'd have our main title up there. Number 3, always label the x and y axes and give units. So for example, temperature on the x-axis would be temperature degrees Celsius or temperature degree Kelvin or degree Fahrenheit. On the y-axis you would put time and you'd put in seconds. Make a line graph unless told otherwise, but don't connect the dots. Most of the time, you do not connect the dots on your graph. Instead, do a line of best fit, which can be straight or curved, but doesn't go from one dot to the other. So for instance, if we have data that looks something like this, then our line of best fit would be a straight line that goes right through the middle. Now, if our data looks more something like this, again, we're not going to connect the dots, but we can draw a curved line of best fit that follows the pattern of the graph pretty well. Make sure your data is graphed as large as possible in the space you've been given. What's wrong with the graph? What I'd like you to do now is pause the video and write down 4 things that's wrong with this graph on the right-hand side. Now, you may have had others, but here are the 4 that I came up with. Number 1, there's no title. You'll notice right up here, there's no title written up there. So that's one thing that's wrong. There's no labels on the x or y axis. You can see there should be something here on the y and something over here on the x. Again, there's nothing. There's no units on the x or y. So again, we should have some kind of unit here, and there's not. And the dots are connected. What we want instead is this nice line of best fit that you can see right there. So here's a good checklist for you to keep in mind when you're making your graphs. Graph paper is used for all graphs, not lined or plain paper. Your name and date should be printed at the top right-hand corner of the page. The axes should be spread out enough to fill at least 2/3 of the available space. The title should be located above the graph and should be written in pen, underlined, and provide information that relates the dependent variable to the independent variable. Axes labels should be written in pen, the name of the variable with units in brackets. So for instance, math in kilograms. Scale should be written in pencil. You should have equal mathematical intervals for equal greatest squares. Horizontal and vertical axes do not necessarily have to be in the same scale. Points. Plot using either an X through the point or a circle on the point. And then you should have a line of best fit, usually a straight line through at least 2 plots with equal number of plots above and below the line. We also talked about how you can have a curved line of best fit as well. So what I want you to do again is pause the video and determine if the axes are labeled correctly or if they're reversed. So here are the correct answers. Check them against yours and make any corrections as necessary. Number 8, discussion. Results are discussed and interpreted. Sources of error are identified, discussed in terms of how the results could have been affected, and suggestions of how to improve if the experiment was repeated. Do not say human error, and be very specific. It's written in past tense and third person, and you're going to state whether your hypothesis was accepted or rejected based on the data. So in general, this is how your lab reports are going to be structured. You're going to have your introduction, your hypothesis, variables, materials, procedure, results, and discussion. And that concludes our lesson on the scientific method.